in biostatistics in 1988 at the University of Barcelona. And after that, he moved to the States and worked in prestigious labs, including the Dana-Farber Cancer Institute at the Harvard University, where he collaborated with Temple Smith, a pioneer of theoretical bioinformatics. In 1994, he came back to Barcelona, where in 2005, he joined the Center of the Regulation Genomica, where he coordinates the bioinformatics and genomics program and collaborates with University Pompeu Fabra. He hired several students of the Bologna Master into his PhD programs, and we are really thankful for this. And now, as to his science, is a leading science in the field of computational genomics, interested in the information process, leading from DNA to protein sequences, and more recently to the investigation of endophenotypes, specifically histopathological images in the interpretation and the framework of cellular and molecular phenotypes. Authors of more than 300 publications in prestigious journals, we are really glad to welcome back you in Bologna, although virtually. So thank you for coming and for participating, Roderic.
Well, thanks a lot, Rita, for your introduction. Let me just one thing. Let me share my screen. I have to go here. See, share the screen. Where is this thing? Share the screen here, and then share this. So you see my presentation right now? Yeah. Can you? I assume that you see my presentation. So
yes.
I really have to say I'm really sorry to be here. And it's not because I am in a bad place. I just took a picture of the view from my office, so you could say, well, that's a great view. But I really miss not being in Café de la Corte. Shit, how can this go back? 1 second. Previous. Why is this not going back? Anyway, so what I was saying is that I'm really sorry not to be in Bologna in Café de la Corte, because this was the opportunity to visit a beautiful city, to interact with you, with the students, and of course see our friends, Gigi, Rita, and others. And one of the things that I really miss one of the really negative consequences of this COVID thing is that for the last 2 years we have not been able to visit you. And I hope that what is this? Okay, because I got some messages here. I see that's the problem. Anyway, so yeah, that's what I want. I hope that next year I will be able to be in person in Bologna. So I don't understand why I cannot move my. Let me just and show 1 second and start again. See whether this works. Yes, okay. So yeah, sorry. Actually, I realized that I have given a title that's slightly different of the title that I gave to Gigi a few months ago, and I still the content of my talk will be very similar. So I will move from the Human Genome Project to what I believe is now one of the most important projects in the history of biology, and I'll give you on the way. This will be very general, but I will try to touch on some of the work that we are doing, in a sense, trying to highlight the challenges that the data that's coming from research in biology it's generating. So this 20 years ago, actually now 21 years ago, I believe, the draft sequences of the human genome were published. So these were not the first genome sequences to be published. There were maybe 2 or 3 model organisms, the sequences of which were sequenced before, but this was really a landmark in the history of genomics and the history of biology. This was really a long process that took less than anticipated, because it was planned to last for 15 years, but actually it ended up in 2001, maybe 11, 12 years. because of the competition that the private companies, Celera Genomics, generated by starting their own parallel genome project in competition, as I said, with the public consortium. Actually, that's the reason why maybe some of you wonder why there are two human genome papers published exactly the same day in the two journals, the most important journals, Nature and Science. And actually, they were, as you see, Science usually or used to get out one day later than Nature. But in this particular, for this particular issue, the two papers were published exactly the same day, so that nobody could claim that one of the projects was ahead of the other. Anyway, this was a project that had a lot of international impact and societal impact. Actually, it was announced simultaneously by the President of the United States at that time, President Bill Clinton, and the Prime Minister of the UK. Where is he? I don't see now. Well, anyway, I thought I had a picture of. Yes, Tony Blair here on the. Sorry, because I have the Ziploc in my screen, and here we have Francis Collins, which was the head of the public consortium, and this is Craig Venter, was the president of the company. Anyway, it was partially because of the Human Genome Project. Was partially because of the Human Genome Project that we had this explosion on our discipline of bioinformatics. Right? That's the number of papers that in Medline in which the keyword genomics or bioinformatics appears, and you can see that, of course, I couldn't do it after 2010 because this program ML Trends doesn't work anymore, right? But anyway, as you can see. Still in 2012, the number of papers with bioinformatics and genomics was less than the 5 or 6 or 7% of papers in Medline that actually deal with cancer. But the explosion is spectacular, right? The explosion is spectacular. It is something that has continued. It has to do, of course, with the explosion of data that is being generated in all scientific disciplines, and in particular in biology, that has to do with essentially has to do with the automation of data obtaining methods. So the data is now being generated without the need of human intervention. So just for you have an idea, of course, I did my PhD many years ago, maybe I started 40 years ago, and I already did my PhD in theoretical and computational methods, but most of my friends and colleagues that were working on the lab, so they spent 3 or 4 years and most of their work was to actually obtain data, right? That they record in a book. It was not an electronic book, it was a handwriting book. So maybe at the end of the thesis, after 3 or 4 years, they have like 5, 6, 7 tables of data. Now, of course, in 1 minute, we generate orders of magnitude more data than that was generated just 20 or 30 years ago. And the effort in biology has moved from effort into obtaining the data to effort into analyzing the data, because the data doesn't need humans anymore to be produced. So what happens after? So there was this big splash about the Human Genome Project. Why was this important? What this will change, not only biology, but actually medicine and maybe even the way in which we live on Earth. So there were many claims that actually have not been fulfilled. I mean, there were serious claims by serious scientists in 2001 saying that in 10, 15 years, 20 years, humans will live 120, 130, 140 years without diseases and things like that. So we actually were over-optimistic on the impact and the short term that the Human Genome Project would have on biology. This doesn't really mean that the human genome or the sequences of the genomes doesn't have an impact in biology. They have an enormous impact on biology. But biology is something we have learned is that biology is much more complex than we thought in the pre-genomic era. So in 2000, this was 2001, 2002. In 2002 and 2003, there was debates on how genomic research should continue after we had the Human Genome Project. Because of course, the Human Genome Project was the culmination of a project. It was the end of a project, but the end of a project is the end of something, right? It's just the beginning of something else, right? The human genome was the foundation of what is coming after in biology. And essentially, if my view of things, there are three major directions in which genomic research has moved after the Human Genome Project. First, the Human Genome Project, the sequences that were produced in 2001 were the single sequence, the sequence of one individual. Actually, it was not even of one individual; it was just a chimeric sequence of many individuals. At least in the public project, in the Celera, we don't really know how many individuals or whether there was only one individual that was the genome of which was sequenced, right? But I think that maybe were 5 or 6 in the Celera. I actually don't recall. But the thing is that we had just one genome sequence, and of course, let's say most important things for human biology are in the variation, right? So what it was soon understood is that without having the understanding how the human genome sequence varies within human populations and how this variation correlates with diseases, the value of the human genome was limited. So one of the directions was to actually try to catalog the human genome variation across human populations, right? The second direction in which the post-genomics project moved was into trying to understand genome function. So we all know, or we knew, that the genome is actually the set of instructions that determine the biological traits in the human and other organisms. But the way in which these traits are encoded or function is encoded into the human genome was mostly unknown. And actually, it turned out to be much more complex than we had assumed. So there has been a large effort into trying to understand how the genome functions. And the third direction in which genomics has moved is into sequencing other species, right? The human is only one of the millions of species that inhabit the Earth, and as we will see, and as we are increasingly understanding, I mean, there is no other way to understand human biology than through comparative methods, right? So to see how it's actually a little bit the same thing that's a little bit the same idea, and I really want to emphasize this idea. There is a parallelism, right, between sequencing human genomes and correlate the variation in human genomes with traits that vary within human populations, let's say age, eye color, hair color, the predisposition to suffer particular diseases, etc., and the variation of the genome across all the species and how this correlates with the traits that characterize this species. In a sense, these are sort of analogous problems, which I think has not really still well understood in the community. That we will soon face a huge amount of information about a huge number of species, and that there is an analogous problem to I emphasize this, to correlating genome variation with traits within populations and between species. And just before I start with going over each of these areas, I just want to emphasize something that is also very important that we shouldn't really downplay. And you have seen, and you will see this probably many times. This is possible, right? These advances in science are possible because of the technological breakthroughs. So without technological advances, progress in biology is not possible. Right? In a sense, what these machines, these are the sequencing machines. In a sense, the sequencing machines are the microscopes of the 21st century, because they allow us to see life at the smaller scale, right? The smallest scale possible. Life starts when the nucleotides get associated, linked into the sequence of the genome, right? Below this is chemistry. So as Leeuwenhoek, I think in the 17th or 18th century, right? I think it was invented or used the microscope to see things that had not seen before, like unicellular organisms. Now with these machines, we can actually see things that we have not seen before. We can see, as I said, life at the smallest of all possible resolutions, the molecular level. So, so let me see what has been done since a human genome project in terms of surveying human genome variation. So let me first state the following. So the Human Genome Project lasted 10 years, and it involved hundreds and thousands of scientists all over the world, and it had a cost of $3 billion, right? It was obvious that it was impossible, right? It was impossible to see. Let me see if I can see the chats in case there are questions. Okay, do it here. So, as I said, it was really impossible to sequence a second genome right after the sequencing of the first one, because it was too costly. So it took years before technologies were developed that would make possible to actually sequence more than one genome. And the initial surveys of human genome variation were based not on sequencing the genomes, but actually into targeting or monitoring specific positions in the human genome through arrays. Anyway, when the technologies were possible, so there was this project called the 1000 Genomes Projects, which actually attempted to provide sort of more representative view of the variation of the human genome, not so much focus on the individuals that had European ancestry, right? Because assumptions that are based in genomes obtained from individuals of European ancestry do not necessarily hold for the individuals that are of other ancestries. And this is one very important issue that we need to take into account when we do clinical trials, in which we try to associate genomic variation to particular traits, right? In what's called genome-wide association studies, that I will talk about this. Because when you find an association between, let's say, a particular change in the genome with, let's say, a condition, diabetes, right? And we have actually inferred this association by monitoring only a cohort of European individuals, maybe only males. This assumption that this position is associated to this condition may actually not hold in other cohorts of non-male, non-white humans. So I think that's something it's actually very important. It's actually now widely recognized within the community that you actually need to survey ethnically diverse populations when you want to really have an objective view or a view that benefits all of the associations between the changes, how changes in the genome sequence actually impact human traits. So as the methods for genome sequencing were advancing, so this was then possible to actually start monitoring not with an unbiased way the genomes of the populations, but actually it was possible to look at the specific conditions and specific diseases. So one of the most important projects at that time was the International Cancer Genome Consortium, in which actually this was based in countries. So countries with focus on particular types of cancers, and the genomes of the cancers and the normals, when possible, would be sequenced in a cohort of, I think, the minimum was 500 individuals, as you can see. For instance, in Italy, focus on rare pancreatic tumors, Spain in chronic lymphatic leukemia, some other countries in other cancers. United States, of course, and United Kingdom, as usual, took the lead with sequencing many cancers. And actually, this really has had I think, in my opinion, has had a large impact in cancer treatment. And part of the progress in cancer treatment has to do with the ability to identify biomarkers for particular diseases, and these are mostly based on changes that occur at the genomic level, either in the somatic genome or on the cancer genome. So this is but not, of course, not only in cancer. In many studies were done in many other conditions and diseases. So this is not always, as I said, not always based on whole genome sequencing, because this was and it still is somehow expensive. But there were methods to sequence only the fraction of the genome that codes for proteins, for genes, which is about 2% or less. It's called exome sequencing. This is much cheaper because it's 2% of the cost of sequencing the entire genome. Of course, a little bit more because you have to do something to capture the regions that you want to sequence, or simply by using what is called SNP arrays. So these are arrays in which positions that are known to vary between individuals are monitored, and the exact nucleotide at that position, but only at that position, of course, these panels, these SNP arrays, have hundreds of thousands of positions. It could be determined, but of course, hundreds of thousands of positions is nothing compared with the 3 billion positions that the human genome has. And this is a map of what is called genome-wide association studies. And as soon as there were, I mean, as you can see, there is a large number of associations, so like 140,000, and this is 3 years ago, variant trait association. So this means positions in our chromosome that are associated to an increased risk of, in the case of diseases, of an increased or decreased risk of exhibiting this, or this disease manifesting in the individual, right? So one of these variants may be position 3,287 of chromosome 3, may be associated to some metabolic disease or something like that, right? So this has led to what's called personal genomics or precision medicine. So projects that, so until now, more or less, all this research, all this sequencing of the genomes has been based or has been motivated by research questions. But as the information of the sequence appears to be more and more relevant in the clinical setting, either for diagnosis or for treatment, I think that what has happened is that many countries have initiated projects to sequence the genomes of their citizens, not initially because of research purposes, but actually to provide better healthcare or more economical healthcare, or using the healthcare resources in a much more sensible way. And these are a number of projects all over the world. The most advanced country in this is the UK, with Genomics England. I mean, this is already all. In the UK, I think that they are close to sequence now, I may be wrong, right? Maybe 1 million citizens, and include these their genome sequences in the medical records, right? So this is essentially where this precision medicine is moving. So in using the genomic information together with, right, together with the other typical information that is kept in the medical records, like X-rays, NMRs, any sort of blood tests, other sort of tests, and because it is becoming evident that by including this information, you can provide better healthcare, as I said, either at the time of diagnosis or at the time of treatment. Yes, and I wanted to say something. Anyway, so this is, and of course, this is essentially, that's what I wanted to say. Essentially, there are two areas in which genomic sequence is particularly relevant. One is cancer, because we can identify the mutations that occur in the cancer genome, and then maybe had some idea on how would be the best treatment for this particular condition, or on rare diseases. Right, rare diseases is another area in which genome sequencing is really important, because you can actually identify, in the case of rare diseases, the mutation. It has a large impact, right, in the individual, and therefore you can maybe, right, by knowing the gene that is affected, in particular if it's this Mendelian diseases in which just one mutation causes the disease, you may be lucky and have some idea on how to treat the patient, right? Because for most conditions, right, for most common conditions, there are not Mendelian. These are not Mendelian diseases. They are caused by the contributions of hundreds, thousands, maybe hundreds of thousands of positions to the genome, and each of these positions contributes marginally to the condition, and it's very difficult, right? It's very difficult to find really treatments that work for this complex common diseases, diabetes, heart condition, Alzheimer, etc., etc. So this represents a problem. This represents a problem because of the following, right? I think that you are all familiar for GWAS, but just let me go over this. So a way in which we can the way in which the GWAS studies work is straightforward, right? Let's essentially what they do is that we have individuals with a particular condition and individuals that are controls that do not have the condition. We sequence the genome of the individuals with the condition and the sequence of the individuals without the condition, the controls, and then we see which changes occur or tend to occur more often or less often in the individuals with the condition compared to the controls, right? And it's very simple. For instance, this is a silly example because this is not the case, but let's assume. But I think for you to understand what I mean, let's assume that we have 1 million individuals that have the blue eyes and 1 million individuals that have brown eyes, and we sequence the genome of these individuals, and we see that in a particular position in our genome, let's say in some position in chromosome 7, all the individuals that have the blue eyes have an A in that position, and all individuals that have the brown eyes have a T in that position. We may not know what is the reason why the A is responsible of the blue eyes and the T of the brown eyes. We may not have no clue of the biochemical evidence, of the molecular evidence, of the different levels between the DNA and the final trait that's shown by the organism. We may not know anything about the biology, but actually this provides strong evidence that this position, right, that this position is associated with this condition. And without seeing the eyes of a person, just by looking at this position in the genome, we can have actually quite good prediction of what is going to be the color, right? So this is the basis of all these association studies. Only that, as I said, this never happens unless this is a Mendelian condition, which is caused by the mutation of a particular gene. What typically happens is that individuals in 51% of the individuals with diabetes, we will have an A here, right? And this will be only in 48% of the controls. right? Something like that. So it's minor changes in the allele frequencies that are associated with disease. And it's the concerted actions of hundreds of these variants that at the end produce the disease. So why I'm explaining this? Well, I'm explaining this because the more sequences we have, the most powerful are our methods to detect these associations, these minor associations, right? To detect this association, which is always, black and white, maybe with 500 individuals, we'll say we'll have enough, right? If I have seen 500 individuals that have blue eyes and all have an A, and 500 individuals with brown eyes that all have a T, I mean, maybe I don't need them. And this is a huge statistic. They have a huge statistical power to claim that there is a strong association. But if the differences are 49 to 51%, I really need a very large cohort, right? And these large cohorts, right? These large cohorts are being now obtained as part of these personal precision medicine projects all over the countries. But these sequences are not obtained for research purposes. So most of these sequences will not be released to the normal way in which the sequences, the genome sequences that are used for research purposes, that actually need to end up in a paper, and then the conditions for publishing the paper are usually that you have to release the data, and the data that is, the data, of course, data that has to do with genomes of individuals is not as free as the data, as other types of data, right? So it has to be managed through specific ways. And for instance, in Europe, there is a database called EGA, the European Genome Phenome Archive, that actually tries to, is the repository of all the human genome sequences that have been used for research purposes. But now this is a minority of all the human genome sequences that are being obtained. There are now, maybe it's 1 or 2% of all the human genome sequencing that ends up in public databases. Most of the human genome sequences will stay within the countries where they are obtained, because many reasons, because political reasons, because also there is a concern on the citizens about the privacy of this data. So this is a very important topic, because this emphasis in privacy of the data is actually to some extent making research much more difficult, because the more genomes we have available with more information about the phenotypes of the individuals that carry the genome, larger our power to detect how variations in the genome impact human conditions, including diseases. So in order to actually deal with this issue, so how can we actually conciliate these two things that we want, right? So the fact that we want to have as many sequences as possible freely available for research, but on the other hand, these sequences are more and more obtained for clinical reasons, and people are concerned about privacy, and countries don't want to share the data outside of their national boundaries. So this is because this is why this organization, the Global Alliance for Genomics and Health, was created, right? So that it attempts to, while maintaining the privacy of the data, make this data as useful as possible for medical applications. And for instance, one topic in which the Global Alliance is working is what's called beacons. So that's the following, right? I'm saying that, so you may not need to distribute the genomic sequences for this to be useful. But let me say, my office, there is a hospital. Let's assume that there is a patient with some rare disease that the doctors are unclear what is the disease and what is the treatment, right? And they sequence the genome. They look at the genome and they see that the genome has mutations in particular positions. And then they say to the world, through this software, through these pipelines, through these beacons that the Global Alliance is developing, is there any other hospital in the world that has seen a patient with these particular mutations? And maybe hospital in Osaka in Japan actually seen a patient with that particular mutation. And maybe they have employed some treatment that has actually been beneficial for the patient, right? So the hospital in Osaka responds, yes, we had seen this mutation, this patient with this mutation, and we have started this treatment, and this treatment has worked. And that's a little bit the idea behind this global alliance. So try to, while respecting all the political constraints associated with genomic data, make this data still useful for the citizens. And so that's why, as I said, I just wanted to stress how important is not only the genomic sequence, but the phenotypic information associated to this sequence. And this is another really extremely important resource for human biology, which is the UK Biobank. And this is in the UK, of course. And this is a cohort of 500,000 individuals that will be followed with over the years. And all this, and they are, I'm not going to read this, you can see here. So many information is being recorded on these individuals. And the thing is that all these individuals have consented for this information to be public. So all this information, it's available for research purposes. And on top of this, all these individuals will be sequenced. So this will be a great cohort for studies of many different associations between genomics and genomic sequence and conditions. And maybe you heard some of the most controversial findings that people have to use analyzing this data, which is these associations always between genetic variants and, let's say, academic achievement, right? So this needs to be always taken with a lot of care because there are many confounding variables, but there are associations that have been found between particular positions in the genome and how well someone does in the academic environment. Whether this is confounded by social factors is something that I'm unable to judge. I have no opinion on this because I'm not familiar with this paper, so I cannot tell whether these conclusions are robust or not. Just one thing that I wanted to mention: many of these GWAS studies are not replicated. So someone finds an association, they use and this has to do with two things: the thing that I mentioned of the bias in the ethnic composition of the cohorts, and the other thing is the small size of the cohorts and abuse of the p-values, right? Until recently. Now things are starting changing. Until recently, most of these association studies could not be replicated. So let's say that a group in Amsterdam found an association between a position in the genome and, let's say, Alzheimer. Then some other group, let's say in Italy, did the same study in a different cohort. In Amsterdam were Dutch people, in Italy were Italians. This could not be replicated. This association between this position and Alzheimer could not be found, and this was very common. I mean, very few actually. The early GWAS studies, there was very few that could be replicated. I think that now, and this is not my field either, I think that now there is an increase in replication of the GWAS studies because the larger cohorts, and also because the more ethnically unbiased the cohorts are. So just to show some of our work that we've done in this area, as I said, we're talking about really large numbers of individuals. So one method that we can one area that we have been working in collaboration with people from the Department of Statistics of the University of Barcelona, and really sorry, but I'm very ignorant about the details of the statistical methods. But essentially, most association studies are univariate in the sense that you, for instance, look at the association with height, right, with one variable. So you have these statistical methods, these regressions, essentially, that correlate, let's say, a particular test for association between genetic variants and this univariate phenotype height or absence presence of a particular condition, or let's say intellectual coefficient or brain volume, etc., etc. But more and more and more, it is really obvious that many phenotypes are of multivariate nature, right? So you need to test for association between one position and multiple phenotypes at the same time. This is, for instance, this is the method that we developed. We call MANTA. This is based on a method that was developed in ecology, which is called Permanova. This is permutational analysis of the variants. This is a non-parametric method it's a non-parametric version of analysis of the variants. And the problem with this method is that require permutations to assess the significance of the association, right? And as we said, the databases and the cohorts grow larger. These permutations were really unfeasible in terms of computation time. And Miquel Calvo, Ferran, and Diego, they found this. They had this really nice theoretical result that they have the asymptotic distribution of the statistic that is computed here, and therefore you can compute the significance in a much reduced amount of time. And that's really very important because actually we could do this largest. Actually, this is one example. We had several examples in the paper, but this is an example using actually the UK Biobank, in which we actually tried to identify variants in the genome that were associated to the hippocampal, the volumes of the several hippocampal regions, subfields, right? These are, some of them are here. And we actually found 45 positions that are associated with variations in the relative volumes within the hippocampus of these regions, which were compared with 10 that were previously known using univariate approaches. So this is, for instance, one such example, right? Of course, as I said, the effects are small, right? These are. I mean, just for you to understand this is the residual volume, whatever. This is the volume. Well, it's not the volume because it will be zero, but anyway, this is related to the volume, and we see that of this particular region. Let me show you what this means. In this particular region, right? We see that the individuals at that particular position, I don't know which one, this is the number of individuals, had this genotype, had a larger volume than the individuals that have this genotype, GC, here, and the heterozygous had an intermediate volume, right? And this actually, this also affected as well, this SNP, this change affected the volume of other regions, right? So this is, I mean, there is, I believe that there is a lot of work to be done in this area of trying to correlate genetic variants with phenotypic traits. And as we move from univariate to multivariate to multidimensional traits, I think that although I haven't really shown any example, I'm pretty sure that people are already working to using machine learning methods to address this problem, because really traditional statistical methods have limitations. Although we have attempted to overcome some of these limitations, I think that there is a room here for this type of methods. So let me go now into the second. I don't know how do you want to do this really. Should I stop for questions or should I stop at some point, or should I have the questions to the end?
My suggestion is that we keep the question for the end.
Okay, good. Okay, so then, okay, so I'll move to the second. The second area of the second direction in which the genomic, the post-genomic projects move, which was trying to understand genomic function, right? And probably the best known of these projects is called ENCODE, which means the Encyclopedia of DNA Elements. So ENCODE It's a project that started right after the human genome. So it started in 2003, and actually it has just ended last year. So it has lasted for 20 years. It's the longest genome or genome project that, in terms of duration, ever. Right? It's the Human Genome Project actually lasted less than the ENCODE project. We have participated in the project since the pilot phase, first phase, and that's amazing because we have seen how the technologies have changed during the year. So in the pilot phase, just for you to give an idea, only 1% of the human genome could be analyzed because the technology at that point, I'm talking about 2003, did not allow to analyze the entire human genome. Anyway, now the technologies have changed. We have now many different assays that tell us how the genome works, from technologies that are able to tell us what is the methylation state of the human genome, or the epigenetic state of the histone modifications, how actually the different regions of the genome are interact into and form the structure of the chromatin, which regions are open in the particular condition, so the transcription factors can bind, and of course, which transcripts are being made, actually how these transcripts are also modified. So this is called an emerging area called epitranscriptomics, which proteins bind to these RNAs, which RNAs are being translated, etc., etc., etc. So we now have a huge amount of information about how the genome is working. And I just wanted to say something on this topic. I am only emphasizing large-scale genome projects. So these are projects that involve hundreds of scientists working collaboratively. But this is not the only, and maybe not even the main contribution to this area. Many individual laboratories have made many relevant discoveries and contributions, right? So don't right? Maybe I'm afraid that I will give the impression that this research is only possible if you do it into these large-scale genomic projects, but that's not the case, right? I think, these large-scale genome projects, in a sense, led the way. Initially, what ENCODE was doing in 2003 could only be done by large consortiums. Now, right, anybody can do a ChIP-seq experiment or a RNA-seq experiment or a proteomics experiment, which many years ago were actually privative of only large-scale consortiums with large amounts of resources. So I think this is one of the values of these projects, right? That they are able to streamline, to develop the pipelines, to decrease the costs of all steps in the process, from the experimental to the computational, etc., and then making this available to all individual researchers. Well, as I said, I just emphasize one project. It's called ENCODE here, right? But there are many. There have been many during the years. Many other projects that aim to similar goal, right? To identify which regions are functional in the genome under which conditions, and that's, I think, also important. The genome is always the same. This computer, the genome is always the cell. In all the cells, it's the same, but the cells are different because the genome works differently in different cells. And that's what these projects are trying to see, right? The genome is static, but the function of the genome changes the cells. And these functional genomics projects are trying to see which regions are active, working in the different cell conditions. So another project that is actually very long, maybe even longer than ENCODE in a sense, is Phantom. This is a project that was initiated in Japan and was based on technology, actually was mostly oriented to identify all transcripts into the human genome and how they are expressed in different conditions, using a technology that was developed at the Roderic Institute in Yokohama, which is called GAGE, Gap Analysis of Gene Expression, which actually allows to very efficiently target the 5' end of genes, and then it allows to identify the promoter region, the most important region involved in the regulation of the expression of the gene. And they have employed this technique in hundreds of different cell lines. Blueprint is like an ENCODE project. This is an European project that is focused on the hematopoiesis. So essentially, what it was monitored was just the cells during the blood differentiation. And I will talk about the GTEx. Is another very important project in which we have all been involved. Next, and of course, these projects as the technologies have progressed, while before we could actually all these projects, all these assays require the use of millions or tens of millions of cells, right? So when you do a bulk RNA-seq experiment, you had a mixture of many different cell types, and what you get is not the expression of any of. It's not the when you do an RNA-seq experiment. I'm saying in a particular typical RNA-seq experiment, you extract the RNA from millions or tens of millions of cells. This is, for instance, from liver, right? You have cells of many different types, so you don't have the expression of the cell of a particular type. You have an average expression of many different cell types. This is what is actually being sorted out through the single cell methods that are working now quite well for RNA, and now they are being a lot of research is being done into making these projects also, these single cell methods also possible for other types of genomic assays like ChIP-seq, ATAC-seq, etc., etc. And there is this huge international project called the Human Cell Atlas that aims to actually characterize from the transcriptional standpoint all the cell types in the human genome. So in the human genome, there are around 10 to the 13 cells. We don't know how many different cell types. This is also very arbitrary, but the aim of the Human Cell Atlas is to try to characterize from the RNA standpoint, right? All the types in the human genome. So I like to. So as we have seen, we've seen this direction in which we have the aim of characterizing genomic variation, and then the other projects we try to characterize genome function, right, by working on the genome of reference. But of course, what we are really interested is in how the genome variation in the genome, right, variation in the genome impacts the function of the genome, right? Because that's essentially what's underlying the phenotypic differences, right? So we want to see why, when we have a change in the genome, right, this particular element works differently in a particular cell type. For instance, why, when we have this mutation in the genome, this gene expressed differently in a particular cell line, right, or a particular cell type. And this is a little bit what the GTEx project was about. The GTEx project was actually, I think, one of the most relevant projects because of the difficulty of obtaining the data. So this is a project in which organs and tissues were collected after the death of the individuals. There were about 1,000 postmortem donors. The organs had to be collected within 24 hours because otherwise issues that related to degradation could impact the biological state of the organs. And then the RNA was extracted from these organs and RNA-seq was performed. And also the genome of these individuals, was sequenced. I don't have the slides here, but since this is about machine learning, it's a pity because I could have brought this. One thing that we were really particularly interested in exploring is the impact of the postmortem interval in gene expression, right? So because we had individuals, the organs were extracted during 24 hours after death, so we could actually see how the RNA change expression in organ-specific way after death, and by using I think it was random forest or some other machine learning method, we develop a method to predict the time since death based on the expression of the genes on organs taken from cadavers. And this actually was quite well. This was had quite a big impact in the forensic field, by the way. We are totally unexperts in forensics, but actually it opens some sort of an area of research, because now this is being, I think, if I am not correct, this is being taken sort of seriously, right? That when you, I mean, this is, you know, that in forensics there is an issue that when you find a cadaver, you want to determine when this person died, and usually this is based on body temperature and what's called rigor mortis, the stiffness of the muscles. But these methods have actually some inaccuracies. And what we propose is that by actually taking RNA from the skin and from the muscle, that are organs that are readily available, and by seeing the expression of the genes, we could actually predict how long was that this person was dead. Anyway, one thing that's important also on GTEx is that there are these histological images. So each of the samples, so at the end, sorry, I didn't mention this, but we had about 20,000 RNA-seq samples from 1,000 individuals, on average 30 organs per donor. And essentially the aim of this project is to understand right how the change in the genome, right? The change in the genome affects the expression of a gene. For instance, we have this gene, right? This is the expression of a gene, and we see that these individuals that have the GG genotype have the expression of this gene at much higher levels, in particularly in the brain, than the individuals that have the AA genotype, right? And the heterozygous have an intermediate expression. This SNP is called an expression QTL, right? It's a genetic variant that impacts the expression of a gene. And in this particular case, the genetic variant, right, impacts the expression of the gene only in the brain. In the other tissues, the gene is not very highly expressed, and this variant doesn't have any impact. Yeah. So for each of these 20,000 RNA-seq samples, we actually had histological images from the sections from which the RNA was extracted. And this is actually very important because it allows also to, as we will see, to correlate changes in gene expression, right, with morphological or anatomical changes in the tissues and in the organs. And this actually could be, for instance, important to predict particular diseases. For instance, in this particular case, right, this is an artery, and this is all these histological images are reviewed by pathologists, and in this particular case, this artery shows symptoms of severe atherosclerosis. So if this atherosclerosis is somehow reflected in the gene expression, and this could be detected, for instance, in the blood, that could be really a good way of making diagnosis, right, of the conditions. So let me just now focus on some work that we have done just as an example of which is actually the intersection of these two projects, the ENCODE and GTEx, just as a flavor of what we do and how we think this is important, right? This is was published in Genome Research. Actually, this is the cover of the Genome Research issue in which our work was published. This more or less reflects this idea that this is based on a painting by Pablo Picasso, and essentially this is I think this is called Girl Before a Mirror. Yes, and essentially this wants to emphasize wants to reflect it's like a metaphor of the changes in gene expression that are associated to disease. Anyway, so what we did in collaboration with the group of Tom Gingeres, I don't know why Gingeres is not here, from the College of Harvard, was to monitor the transcriptome of primary cells. These are cells, these are not single cells, but these are a group of cells that are in principle quite homogeneous from different body locations, right? That's what we did. And we did RNA-seq, and then we clustered, we just compare the transcriptome of this, I think it was 53 or something, different body locations. What we did is we just compared the transcriptome of these primary cells. So that means the gene expression. So we had a vector of 20,000 values, in which each gene we have the expression, and then we just compared all against all, and we did the clustering so that when the transcriptomes are more similar, the cell types cluster closer to each other. Of course, the diagonal is 1 because we're comparing a cell against itself. And as you can see, there is just visually, you see big, like 3 or 4 big clusters, right? And when you look at these clusters, they do not, this is the body location, right? They do not correspond to the body. We could say, well, maybe these are maybe all the primary cells that came from the stomach, for instance, right? And these are the ones that came from the liver. That's not the case. So this clustering doesn't really respond. This doesn't really respond to or correspond to body location. Doesn't correspond to developmental. We could say, well, this has to do with the origin, with the embryonic origin of these cells. This is not the case either. Essentially, what we found is that the cells belong to a variety of types, like include fibroblasts, adipocytes, muscle cells, mesenchyma, stem cells, etc., etc. We call this mesenchymal. Then we have another group of endothelial cells and another group of epithelial cells, and then we have these melanocytes that cluster separately. And when you look, for instance, at a particular organ, let's say lung, for instance, you will see that the cells from the lung are everywhere, right? The cells from the lung are everywhere, so they don't, and this happens with all the organs. I think that's an important thing. So this means the following, that I can take a cell from the bone of my foot, for instance, and two cells from the lung, right? 2 cells from the lung, and 1 cell from the lung may be more similar to the cell of the bone from my tooth than the other cell of the lung, right? So there is a commonality of cell types, the commonality of cell types that do not really correspond to tissues or to organs, but actually are what we call the basic components of the cells in the human body. We actually came to the conclusion that there are 5 major cell types in the human body, in addition to these 3 that I mentioned to you: epithelial, endothelial, and mesenchymal. There is we analyzed other data types. I didn't show this because we didn't monitor cells from the blood or from the brain, but we saw that there were another type which that corresponds to the cells from the blood, and then another type that corresponds to the cells from the brain. And actually, this is more or less corresponds to the histological types that maybe you still learn in school. I don't know. These are the traditional cell types in which tissues are typically classified. I mean, typically endothelial is considered a subtype of epithelial. From the transcriptomic standpoint, we don't see this. Endothelial and epithelial are 2 separated cell types. Blood is considered a subtype of connective tissue. We don't see this, but it's type by itself. And actually 2 types of tissues that are considered separated, like connective and muscle, we think that they correspond to the same type, which is mesenchymal. And we actually try to see whether single cell data that became available could actually support this hypothesis. And indeed, this is from mouse. Indeed, we actually found that these are about 50,000 cells, I believe. Yes, 50,000 cells, more than 50,000 cells, for which the transcriptome of each cell was sequenced. And we actually see that the cells from the blood, the cells from the mesenchymal cells, the endothelial cells, and the epithelial cells, and actually the hepatocytes cluster very separated. Hepatocytes, I think, are typically considered epithelial cells, It's sort of epithelial, but very, very, very differentiated epithelial cells. And actually, when we do the clustering, we actually see of the clustering of the different cell types derived from the single cells. We actually see that they cluster more or less into our major cell types. So we identify genes that are specific of each of the cell types, epithelial, endothelial, mesenchymal, also from the blood and from the brain. And what we did is we use methods that allow to what's called the convolution methods, that based on the expression levels of these marker genes can attempt to estimate the cellular composition or the proportion in a particular bulk RNA example of cells of this particular type. Let me say what I mean. So we have a sample from the liver, right? It's bulk RNA. We don't know which cells are there, right? But we only have the expression in this mixture of cells, right? In this mixture of cells. The cells may be of very different types. So we claim that there are 5 types. And there are methods that based on the expression of these marker genes, right, can estimate what is the proportion of this liver cell of epithelial, endothelial, mesenchymal, melanocytes, or whatever, right? So let's say, well, we estimate based on the expression of the genes that 50% of the cells in the sample are epithelial, 30 are endothelial, and 20 are mesenchymal, or whatever, right? Anyway, I will not go into the details. This is we use a method called Excel at the end. So this is our estimates of the enrichment. This is cost of enrichment of the different major cell types in each organ. Of course, as expected, all the neural tissues were enriched for cells of neural type. The blood was enriched by the cells that we consider blood cells, right? So, and in general, things made a lot of sense. For instance, in GTEx, we did this decomposition using the GTEx dataset, right? So for each sample in GTEx, we did this decomposition, and here is the results of this decomposition per organ, right? Per organ. So we infer these types in a code using these primary cells. Then we infer the markers of these 5 cell types, and we use these markers of these 5 cell types to estimate the proportion of these cell types in the GTEx tissues, in the 20,000 or whatever GTEx tissues. And this is the distribution of these estimates in these samples from GTEx. And the only thing that I wanted to mention is that these estimates make sense. From GTEx, there are 2 types of esophagus samples, one that comes from the mucosa and one that comes from the muscularis layer. So the esophagus has 2 layers. The external layer is an epithelial layer, it's the mucosa, and the internal layer is mostly muscular. And we see that in the mucosa, we see enrichment of the epithelial cell type. In the muscularis, we see enrichment of the mesenchymal cell type, as we have predicted. And what we could do is actually, I don't know, this should move, but doesn't really matter. The idea is that we were able to cluster the samples in GTEx based on the proportion of cell types, and actually the proportion of cell types are quite sort of a signature of the organ, right? So just this is based on the proportion of these 3 cell types, mesenchymal, endothelial, epithelial. As you can see, just looking at the estimated proportion of these cell types, we can actually classify the tissues quite well. So this means that this broad cellular composition defines tissue type, and that alterations in this composition may reflect tissue histology, morphology, and pathological states. And actually, to test this hypothesis, we actually went to the histological images. These histological images are actually quite amazing. I mean, let me see if I can go here. Yes, so I mean, this is a test. Let me share for that. Well, I searched this because I learned the cell types, and then now I forgot the names and the cell types. Anyway, so this is what I am showing here. So you can zoom and you can actually see the individual cells. And as I said, I choose this because there were only 3 or 4 types of cells, and I learned what were the spermatids, what were the spermatozoes, what were the whatever thing, but now I forgot. So I cannot tell you which cells are which, but some people expert in cytology, right, could really tell you this. Well, anyway, so the idea that we wanted to see is whether we could relate. Now, for each of these samples, right, we have the gene expression, right, and also we had our estimates of cellular proportion. So now the challenge here, which is a challenge, we've been working on this for many years, is how to see alterations, right, alterations in these images, right, that can be associated to changes in gene expression. This is very challenging. We have done something, but I don't have many results of this. I will not show the results that we have on this because they are difficult, right? We spent a lot of effort with little success, I have to say, right? But we have this thing, right? We have this thing, right? For instance, we have the pathological comments, active spermatogenesis in this particular case. Of course, what we would like to do is, based on the values that we have of gene expression, to predict that in this sample, spermatogenesis was going on. But as I said, dealing with the images is very difficult. And here is where you really need, and this is what we have been doing, machine learning methods, right? To try to extract features from the images and correlate these features with the expression. Anyway, so we took the easier route. So let's forget about the images and work with what the experts have seen in the images. And this looks easy, right? But it is now. Did you see my? Sorry, did you see my?
Yes, yes.
You see, and now you see
the
again.
Pointer.
Yeah, and now you see again the presentation, right?
Yes.
Yes, yes.
Yeah. Okay. But you saw, right, when I zoom in and out from the.
Not really.
Not for the zooming.
I see. Sorry. I was talking, assuming that you were seeing what I was seeing. Anyway, you can do it. If you see this link here on the top, I click here and I went to the browser and I was browsing through the image. I was zooming in and out, and you didn't see this. I'm sorry. Anyway, that's what I was saying. Anyway, what I'm saying is that if you zoom in and out, you could see the individual cells. This is what I was saying, but you were not. I say that this is very difficult to correlate this with the expression or with the cellular composition, and we took the easiest route of looking at the comments by the pathologists. But this is not trivial either, because these comments were not structured, right? Where they were just free text comments, and it was difficult to know whether, if you, for instance, wanted to know whether there was spermatogenesis going on in the testes, it was not enough to search for the word spermatogenesis, but it could be active or could be absent spermatogenesis, or no spermatogenesis, or mild spermatogenesis. And for instance, fibrosis could be severe fibrosis or no fibrosis or whatever. And of course, the doctors make typos and etc. etc. So we actually, the student Manuel Muñoz, actually use machine learning methods to actually parse these pathology comments, and at the end, I will not go into details. At the end, came up with a list of GTEx histological phenotypes that actually is the list that now the GTEx consortium is using. So this is the number of samples that are annotated with congestion, with fibrosis, with spermatogenesis, with atherosis, classification, etc., etc. Some of these phenotypes, right? Some of these phenotypes, of course, are common to many organs. Some of these phenotypes are common, are specific of a particular tissue. And then what we did, now we had, for example, a binary classification, whether it was fibrotic or not, whether it was atherosclerotic or not, and then we could compare whether between the samples that were fibrotic or not, or atherosclerotic or not, we could observe changes in our estimated cellular composition. And this is actually what we found in several cases. For instance, in tibial artery, we found that in the atherosclerotic cases, we had an elevated proportion of endothelial cells compared to normal cells, right? This increase in endothelial cells in the atherosclerotic versus the normal. But for instance, in atrophic muscle, we found an elevated proportion of mesenchymal cells compared to the normal muscle. We actually see interesting histological associations with cellular composition. So the most different transcriptomic, more different organ between males and females is the breast, right? It's the breast, mostly because the female breast is very rich in epithelial cells compared with the male breast, right? And this is the main difference. And this actually, when you do a transcriptomic analysis of the GTEx, you try to find differences between males and females. The organ that stands up is the breast, right? But it's not because the cells of the breast express a different number of genes. It's mostly because the cells of breast are enriched for epithelial cells compared to the male breast. But there are males that are annotated with gynecomastia. Gynecomastia is a condition in which males grow, in particular when they get old, breasts that look like those of women. And actually, you could see that males with gynecomastia had a cellular composition that is more similar to females than to males, and actually mirroring as well the histological phenotype. We also see altered changes in the proportion of different cell types that occurring with age. This is a young lung. This is an old young, and as you see that with age, we see a reduction of the epithelial cells, which is actually consistent with some studies found in mice, where these alterations of cellular composition are particularly relevant in cancer. And we look at the ICCC, and actually we really saw that in some types of kidney cancer, these are the cancer samples. These are the normal samples from GTEx and from the controls from the cancer projects. We see an elevated proportion of endothelial cells, with the exception of this cancer. But actually, in this cancer, it's a different type of cancer in which there is no increased vascularization, because in this cancer there is an increased vascularization that actually explains the increase in cells of endothelial type, and you see it for each individual. You see this difference, right, when comparing the normal, the normal with the tumor. It also, cellular composition can also correlate with the degree of the progression or severity of the cancer, right? So this is the case of ovarian cancer. We see a huge increase in the epithelial composition of the ovary, right? It's very, in normal normal ovaries, we see is really small proportion of cells of epithelial type. In ovarian cancer, we see a huge increase in the proportion of the cells, and actually which it's associated with the severity of the progression of the cancer. Mm-hmm. So this is a way of showing you some. There are hundreds of papers that mine the encode and the GTEx data in one way or another. This is just one, just to give you a flavor of what we have done. We have done by mining the data of these two projects. There is a project actually that goes a little bit further. Is a continuation. It's actually the conjunction of ENCODE and GTEx. It's called the ENTEX project. So in GTEx, right? In GTEx, we only had RNA-seq, right? We only monitored the RNA. In ENCODE, we had many assays, right? We had many assays. We had many RNA-related assays, microRNAs. Then we had chromatin assays, AT-seq, DNA-seq, methylation assays. Then we have transcription factors assays, histone modifications assays, RNA assays, etc., but not in GTEx. The NTEX project, what it did, it took 4 individuals from GTEx, and they were the genomes of these individuals were sequenced at high accuracy using a combination of technologies, and then about, I forgot how many assays, maybe 10, 12 different assays were performed in 30 different tissues. So it's the first time that we had such a resource in which we had different individuals, only 4, unfortunately. We had 30 tissues, the 4 individuals, and then we have a number of genomics assays. So we can actually investigate how the relation between the epigenetic and the genetic and the transcriptomic layer, right, changes cell function across individuals. And this is the embryo of the new project. All these projects, by the way, are funded by the National Institutes of Health. Then quote and GTEx, from the United States. I'm not sure I mentioned this to you. Anyway, so now the next, in particular by the National Human Genome Research Institute, which is the institute from the NIH that deals with genomic research. Now, the next big project of NHGRI in this area is called IGBF, which is impact of genome variation in gene function, right? In a sense, tries to link these two things, right? The variation in the genome with the function of the genome, right? The variation across individuals in the genome variation. So I think I am really going, well, I don't know, maybe it's too many things I am explaining. I hope you are not getting lost. I don't want you to get lost on this, because maybe it's best if I don't really, I don't know. I think I will only brief touch. Will touch briefly on the next topic, because I think that I just realized that Montserrat Corominas is going to talk about this as well. So I will maybe just briefly touch on what we have done on the next, but on the final direction of where the post-genomic projects are going. But I just wanted to recapitulate what I said so far. So one of the, sorry, I missed one slide here. Anyway, so essentially the challenge or the aim of biology, or at least of organismic biology, is to understand how the DNA determines the organism, right? And this is very complicated because there is multiple intermediate steps in the path that goes from DNA to the organism, right? So the DNA specifies RNAs, RNA molecules, which are translated to proteins. The RNAs and the proteins organize into cells. The cells organize into tissues and organs. The organs into the systems, and at the end to the organism. And this happens, in our case, during the 9th month of development, right? This is an amazingly complex, from one cell to 10 to the 13 cells, which is an amazing, process. And most of our understanding of how changes in the DNA impact the phenotypes of the organisms are through what I call, what I mentioned, genome-wise association studies. So these are just correlations, right? We don't understand why this variation affects this phenotype. We only see that this particular variation occurs with increased or decreased frequency in this condition, right? And this is what, as I said, this is what maybe this number is different to the one before, because it's a different slide, but anyway, so the other one is more recent. So this is how we still now, right? We still now know most of the associations between the genome and the human conditions. We are unable, right? Let me tell you, and put it another way, there is no way in which anybody can take, as far as I can tell, right? Take a mutation in the human genome that say, well, if I change this nucleotide in this position from A to C, what will happen is this, right? This RNA will be synthesized at different level, then the protein will be less abundant, so there will be less aggregation of proteins. It will change slightly the conformation of the cell. The cell will not be able to adhere as well to other cells, and the organ will have some malformation that will have an impact on the phenotype of the organism. We are unable, right? This is impossible, but we would like to do that. We would like to be able to understand all the path, right, that goes from this change in the DNA to the phenotype. But so far, most of what we know is simple statistical associations. We know nothing about this path, or we did not know nothing about this intermediate path, which is what biology research is. This starts changing when we had microarrays and then RNA-seq, and then we could actually start measuring the expression, right? Measuring the expression of these molecules, the levels of expression of RNA, and this allow us, right? Because when we had values, and like in this text project, right? When we had values of expression and the genome of individuals, we could actually identify the sequentials, right? We could identify variants, right? Variants in the genome that affect the expression of proteins, of the expression of genes, of RNA and eventually proteins. And of course, this starts helping to understand how this variant affects a particular trait, right? Because if we know that this variant is through a genome-wide association study, right? Through a genome-wide association study, it's associated to a particular trait, and we know that this variant also affects the expression of this gene, we could start thinking that maybe this gene, right? This gene is involved in this condition. And the other thing that RNA-seq allowed was actually to the identification, right? The identification on how changes in gene expression, right, affected to identify changes in gene expression that were associated to particular conditions. As for instance, a disease. We have a control, we have a disease, and we see the genes that change expression, in the disease compared to the control. So that we're able now, of course, for more than 10, 20 years, to relate changes in gene expression with the phenotypes in the organisms. What the Human Cell Atlas is doing is trying to bridge the link between the molecular and the cellular level, by trying to characterize the RNA and protein composition, eventually protein composition of all cells in the human body. And through new methods, increasingly sophisticated methods, imaging methods, we are also able to start connecting the cellular world with the tissue and organ world, right? It's through digital imaging of tissues and organs, and also, what I didn't mention here, through digital imaging for sequencing, because we have these spatial transcriptomic methods that allow us now not only to say which genes are expressed in which cell, but where this cell is expressed, where this cell is positioned anatomically in the organ. So I think that there are many challenges into understanding this pathway at many different levels, which require very different expertise. It's impossible to have someone that's expert on GWAS, on image analysis, on single cell sequencing, right? So this requires actually a good understanding of these technologies. I think that on this pathway, right, and this pathway, many relevant research will occur, and many challenges, in particular at this level of the imaging that we have of organs, how to deal with this. And here, methods that are able to, like machine learning methods, that are able to extract features from the images and correlate them with other phenotypes, either molecular phenotypes or organismal phenotypes, are going to be very important. So let me, and I will go very briefly on this because I think that this will be dealt with Montserrat Corominas. So, but just to wrap up and give you a flavor of what we are going, is that there is of course the human is not the only species that that exists on Earth, and there are at least 2 million eukaryotic species that are known, or 1.8 million eukaryotic species that are known. We don't know how many there are. There are estimates that say this 5 million eukaryotic species, 8 million eukaryotic species, 13 million eukaryotic species. Many of them are microbes, right? So it's difficult to identify them. Many of these species, maybe most of these species, unfortunately, we will never know about them because of the massive extinction that we are witnessing and that we are causing in particular. Essentially, there is an interest in actually cataloging the biodiversity of all the species living on Earth, and this already happened, right? As I said before, the human genome, there were a few organisms. The genome of a few organisms were sequenced. I think it was mostly worm and fly, and then after the human genome was sequenced, other organisms that are important for human biology were sequenced. For instance, the sequence of mouse, the mouse genome sequence followed immediately the sequence of the human genome. The rat, right? Everything that can be used to understand human biology was given priority. And then after that, so the genomes of the species that are important for human, for a source of food, like the rice, the mice, the chicken, the cow, etc. And then the sequencing of species that were pathogens for diseases like malaria, etc., right? But these were mostly driven by particular interests on human biology or agricultural interests or applied interests, whatever, right? Now there is an effort, which, as I said, I think it's one of the most important concerted efforts in the history of biology to sequence all the species on Earth. This is, it says 1.5 billion. I don't know. I keep reading different numbers. I guess that nobody knows. Now the number, it's 1.8 million. These are species by no means that someone has given it a name, but of course, whether two things with two different names are the same species or not, this is always complicated. And the aim is to obtain, as I said, close to 2 million eukaryotic species. I will skip this because I think that this will already dealt with the talk of Montserrat Corominas. But there are just to give you an idea, this paper was published like 2 weeks ago in PNAS, a number of papers. I think that this is the proportion of species that have been sequenced, right? For instance, this means that if we take the major phyla, right? Major phyla, so we have at least one species sequence from 80% of this phyla. But as we go down into the phylogenetic scale, right, we see that only 0.40. I mean, we have if we consider the level of families, we have representative for only 15% of the families. And if we move to a species, we have the genomes of only 0.43% of all the species on Earth. So within the next 8 years now, because this started in 2020, and we are already 2 years on the project, so by 2030, right, this should become 100%. This appears really a challenge, but there are many groups working on this. And let me finalize on, I don't know, maybe I should stop here because otherwise there will be no time for questions and you may be already tired of this information. So I will just wanted to say something. So the importance of so a genome sequence with an annotation of where the genes are is really not very useful. So the first task, once you have the genome of the species, is to annotate the genes. This is more difficult than you may expect because I'll give you an info, right? 20 years after the sequencing of the human genome, we still don't know how many genes are in the human genome. right? We still do not know. So there is a consortium called GenCode, from which we are part, that tries to find all the genes into the human genome, and at each release of the GenCode consortium, which happens twice a year or once a year, I don't know, it used to be every 3 months, but I think it's now twice a year, there is a change in this annotation, new genes, and in particular new transcripts, because the genes, right, this is a gene, the gene have many different transcripts, this is another gene with many different transcripts, etc., etc. So these are continuously being discovered as we monitor more and more different cell types. So finding efficient methods to find genes, I think it's really important. And I will not explain the details of our method because it's too late, and I think that you'll be tired. But I think two considerations that are guiding, which are usually not taken into account, right? Which is guiding our work. If you really want to have a good annotation, of the genome, you need to combine a large amount of information of many different types in very complex pipelines. This is sort of available to only large centers. The typical one is Ensemble, right? Is Ensemble. They use complicated pipeline, which is actually quite time consuming, and for annotate the genome, it will take typically months, maybe now weeks, right? But still, it's still a challenge. So we have reformulated the problem to just identify the protein coding genes. The protein coding genes are relatively easy to identify compared to the long non-coding RNAs. And the important thing is that even though there are many, this is very complex. At the end, the work of several groups, I think Alfonso Valencia is talking in this school as well. Essentially, more or less, each gene has one major protein isoform that's always the same. So even though in theory, genes have many alternative splice isoforms, in most cases, essentially in most cell types, we always find the same one, which is the most abundant, right? So we focus our method on to trying, we're working on this very simple problem. And as I said, our guiding principle is to have a method that's very simple, that can be used by anybody, anywhere in the world, right? Because this is this issue of equity, right? Equity means the following, because this is also related with the topic of the bias composition of cohorts. So there is a danger here of scientific imperialism, right? So the idea that, again, the people from the UK and the United States are going to go to all the world, Australia no, because it's already okay, Africa, South America, Asia, they will collect the samples and they will do the sequencing of the genomes. And that's not the way in which science should proceed in the 21st century, right? So what should happen is that this really happens around the places where the species live, right? So that we really empower all the societies in the world at the same level in scientific research, right? So the first guide that we are working is that we want really to empower people that they use their methods at their sites, that they don't depend on a central resource. So our program is trivial to install and to run. We want our program to be trivial to install and to run in any place without any need of sophisticated training of the program and of the individuals. And second, we want our program to be very efficient computationally, because there is a concern, and because you don't think about this, but bioinformatics actually contributes to the carbon footprint of the world, because computing generates a lot of heat, as you know, right? That consumes a lot of resources. So we are trying to have a program that is really very friendly ecologically, and that's our aim. And I think compared to the standard in the field, we are may not be better in terms of accuracy, but we are much, much more efficient in terms of use of energy. Okay, so let me skip all this. This is the people that did the work. This is the people that did the work that I did not present, the work on gene funding. And this is the people that did the work on GTEx and ENCODE. It's mostly the work of Alessandra Reschi. By the way, Alessandra Reschi was a student of your master. She did most of the work that I presented on the primary cell types. This is really a nice piece of work. Alessandra Reschi went as a postdoc to Stanford, group of Mike Snyder, and now she works in a company because that's what she always wanted in the Bay Area. I had other students from your master. Marco Mariotti was the first. He worked actually on something that I haven't really explained here, cellular proteins, which is a topic that I am very interested. And he did, after finishing the PhD in my group, he went as a postdoc to Harvard, and now he's back in Barcelona. I'm sorry he didn't go back to Italy, but the thing is that he met someone, Catalan person in my group, and they are now together. They have a child, and they return back to Catalonia, to Barcelona. And other student that I had, which just finished the PhD thesis, is Beatrice Borciari. I haven't presented anything from her, although she has really very nice results. And she's actually now going for a postdoc in Yale, in the group of Mark Ernsten. I think she's leaving unfortunately, she's leaving by the end of the month, maybe next week or 2 weeks from now. And I just wanted to thank as well in this GTEx project, something that is very important is the people that donate the bodies of their families, the family members, in very difficult circumstances for something that is very difficult to understand. Because if you tell someone that has lost her husband or their child in an accident that you are going to use the body for organ transplantation, you may be able to rationalize the speed of the pain, right? But if you tell someone that you're going to use the organs of this person for RNA-seq, that's really very difficult to understand, right? And these people actually were very generous in this, and something very interesting, actually, the American style is that these people came to some of the GTEx meetings just to see how the organs, the bodies of their people that they love were being used in research. Okay, so sorry because it's a little bit long, but I'll leave it here, and I hope that you all least got some part of what I explained. Let me stop sharing, no? Maybe better. Yeah, stop sharing. Okay.
Thank you very much.
Shit, it's almost 2 hours already. I'm sorry for you. It's too much. I cannot follow more than 20 minutes. 15 minutes is the most I can pay attention. So
No,
I don't blame if you
it has been fantastic. I ask people who have questions to raise the hand from the reaction bar, and I will give the possibility to ask question. First, Professor Casadio.
No, there was somebody else actually that raised the hand. In any case, I will. So it's Marco. No. Okay, that's me. Okay, thank you very. First of all, thank you very much for this. I would say wonderful overview on what went on and what's going on in the field in which you are the expert in Europe, in my opinion. But I have a major question which baffled me a little bit in relation to whatever I think it's a very breakthrough. It's a breakthrough in the field. So suggesting that all these primary cell types are somehow there in all the different tissues. So my question is relative to the following. First of all, how do you recognize the different cell types in the different tissues, and how sensitive is the technique? And then how is it possible, because I'm a physicist, don't forget it. How is it possible that in any case, although being in a different environment, these different cell types do conserve their major characteristics as primary cells?
Mm-hmm. No, that's a great question. So first, we really don't know how many cell types are in the human body, right? Because it depends on what you define as a cell type. We are discovering more and more. There are many types of neurons, for instance, and we are discovering as we advance on these single cell projects, and we do monitoring of the transcriptome of the single cells, and we see all these beautiful TSNE or whatever maps. You see, you recognize the types of neurons that you know, but you also usually find subtypes that you had not before identified, and of neurons of other pancreatic cells, whatever. So we don't know. I mean, I read numbers of cell types that go from 200 to many thousands of cell types, and of course, it depends on where you put your threshold of defining what is a cell type. What we found, of course, we monitored only 50 different primary cells from different body locations, and when we cluster them, we found these 5 clusters, right? We found five major clusters, and we realized that these clusters did not correspond to the organs. So it's not that the lung cells clustered together, the stomach cells clustered together, or the bone cells clustered together. What we found is that there are these 5 types that are found in different proportions in different organs. So in the liver, you find whatever proportion of epithelial cells, of endothelial cells, of mesenchymal cells. Of course, blood cells are found mostly on blood, and neural cells are found mostly on brain, right? But of course, for instance, in pancreatic, we found a composition of neural, because some pancreatic cells also derive from the precursor of the neural cells. So we think, of course, I mean, this will not be really proof until we had more single cell data. So we had a strong problem. I mean, we had really this is sociology of science, and I'm not sure I want to disappoint the students. So this paper that we published in Genome Research was under revision for 2.5 years in Nature. It was sent to 5 reviews, 3, 4, 5, and it was rejected because one reviewer was totally unconvinced of the things. I think he was wrong, but the editor sided with the reviewer and not with us. Anyway, because I agree with you. I think this is a very nice work done by one of your students, Alexander Resky. I think it's really, in a sense, it's redefining the basic cellular types into the human body. And many reviewers recognize this, but one. Anyway, the editor sided with this. Also because this is something that you need to learn. When we started this project, there was almost no single cell data. When we ended this project, there was a lot of single cell data. Well, why don't you use single cell data? Of course, we could not start again with single cell data. And it was really pathetic. It was really very disappointing. It's one of the most disappointing experiences in my scientific life. But you have to learn from the disappointment, because you think that you have this paper already accepted in Nature. You think, well, that's a big thing, right? At the end, it's not. So anyway, so how you're asking how precise are these methods? The single cell methods in principle are quite precise, right? So I do agree that when we will have single cell data from the whole human body, we will be able to build this hierarchy of cell types, and we will see how the cell types organize by themselves. We just were predicting based on bulk RNA-seq. In bulk RNA-seq, you don't really know. You cannot know, right? Because you have just one value for the millions of cells that you have there. But there are these deconvolution methods that, based on genes that are markers, make an estimation of the proportion. We think, because when we look at different cases that we more or less suspected what was the composition, I saw the case of the esophagus, we think that our method worked reasonably well. But of course, it was not perfect. It will only give the broad picture of the cellular composition in these 5 types. But of course, we at the end we didn't even say that this was 70% or 80%. We just produced these enrichments because it was very difficult to say that this was 80% epithelial, 10% endothelial. We just say this is enriched in endothelial, this is enriched in epithelial. And this was one of the big issues that we have with this reviewer, which actually we responded wrong in this sense. So anyway, it was exhausting, this fight with the reviewer and the editor for this paper, I have to tell you. Anyway, this is not the question. This is something, but I think that you should also, if you want to be scientist, you need to learn not only about science itself, but how is to work on science. You have to do it intelligent as well when writing the papers and responding to reviewers. So the answer to your question, when the human cell atlas project is finished, we will be able to see whether we were right or not. Whether really this idea that there are 5 major cell types maintains. I mean, not all cell types belong to these 5 types. For instance, we saw hepatocytes, we saw melanocytes. There are very specialized cells that may not fit this. But I think that we are right when saying that a large fraction of the cells in the human body belong to these 5 major cell types. Maybe 80%, maybe 70%, maybe 90%, I don't know, but the large fraction belong to these cell types, I think.
Very good. May I don't know, do you have other questions or may I go on with another question?
You can go on. There is some question on the chat, but we can go on.
Okay, but if you want. Okay, the other question is relative to the first part of your talk, in which apparently you said that the larger amount of people we have in the cohort in order to do GWAS data or to produce GWAS data, the better it is.
Mm-hmm.
Now, the problem obviously is then related to the how big has to be the cohort in order to come up with reliable data from GWAS. So to say, of course, GWAS is the first step in realizing that some portion of the gene is perhaps involved in some diseases or not, or can be associated to some phenotypes. But then since then later on we are running exome sequencing, or many people does, not us, but we are running exome sequencing. How would you feel about it? So how big has to be a cohort in order to have reliable data out of GWAS?
Yeah. So I'm not an expert on GWAS, actually, although we did this study now. And I believe that the people that are experts on GWAS may have an answer to this. But essentially, the answer is the following. It depends on the effect size. If you have an effect that's very strong, right, and you have and the contribution of this variant to the effect is very strong, right? Let's say eye color, we have only 3 or 4 genes that are I think maybe 5, I don't know. There are very few genes which have a strong effect on eye color. Then you with a small sample size, you have enough, right? You have enough. I mean, let's say the following thing. That's a silly example, but we have the strongest phenotypic effect in humans is sex, right? Males and females. So I think that if you had 10 males and 10 females, and you would find that all the females have 2 X chromosomes and all the males have 1 X and 1 Y, I think that you would conclude that there is a strong association between sex and chromosome Y, right? Because the effect is so large. The contribution of chromosome Y is so large, although it's not 100%, because we know that this male and female thing is not as binary as we assume, but let's assume that it's very binary compared with other phenotypes. Now, if this variant has a very small effect on the phenotype, you need a very large cohort. So, because more complex diseases, right? More complex diseases are caused by many genes with small effects. The larger the cohort, the better. But I think the problem here is the following. Because we are all related to each other, right? Mm-hmm. At the end, most of the genomes are the same at the end. So there was a limit in which the thing is that many variants have never been assessed by evolution. right? Never been assessed by evolution. So we will never be able to find all associations between conditions and phenotypes unless we do, which is now being done, this high-scale mutagenesis experiments, right? In which you systematically mutate, not the genome because it's not possible, but maybe a protein or a set of proteins, and then you assay a particular phenotype, cell growth or something like that, right? This cannot be done in humans because we are not enough humans on Earth for all our genome to have been assayed on the possibilities of the genome. Every new person is essentially a new genome. We never replicate a genome that occurs before, but this new genome is very similar to the genomes that we already have. So it's not really adding an independent. Any new genome is not adding an independent observation. It's actually a quite dependent observation to the genomes that we already have. So it's a complicated. I'm not an expert on this. This is I am just elaborating now here. But I think the short answer is effect size. If the effect size is very large, small. Is with a small sample. If the effect size is very small, you need you use a large cohort to detect it.
Okay, thank you very much. Perhaps what you can do is to read the questions from the chat. Huh?
Yes, I can read for you if you want.
Okay.
The first genome sequencing is done from single genome, or there are multiple samples from different individuals?
Yeah, so this is, I think I alluded to this. So in the case, I think I recall correctly, in the case of the public consortium, there was the clones from the sequences were obtained from many different places, probably coming from many different individuals, right? There was not a single individual that was sequenced. In the case of cellular genomics, which started quite late into the process of genomics by the end of the 90s, I think, if I am not wrong, that is the genome of 5 or 6 individuals that were pulled together. I think that this is, I think I'm correct on this.
Okay, a second question from the same person is, without reference genome, is there any way to identify genomic variants?
No. In principle, no, because you have a reference genome and then you sequence a new genome, you compare with the reference and you see whether there is a change. You could, in principle, without the genome, you could in theory, of course. I mean, you don't have a genome and you have several individuals and you do the short reads. And you mean before assembling the genome, and you could compare the reads from different individuals and see whether there are variations. In the case of the human genome, this doesn't make any sense. The best way to detect variants is to map into the genome. Now, if you have another species, I don't think it makes any sense. Now it's relatively cheap to obtain a genome. I would obtain the genome, and then if I'm sequencing other species, I am sequencing other individuals, map the reads to this. Of course, you could think that without sequencing the DNA, by just sequencing the RNA, you could as well identify variants, right? Because the variants in the DNA transfer to the RNA. But I cannot imagine any case scenario, unless, of course, is in these metagenomics projects in which you are monitoring many genomes at the same time. You may not assemble them, and you may still want to find variants. So it may be possible, right? But essentially, the easiest thing now for a genome that you can isolate, for a genome on a species that you can isolate and sequence the genome, is the best is to sequence the genome first, and then from the other individuals to a low-pass sequencing and mapping into this reference genome and see the variants.
Okay, thank you. Any other questions? If not, I thank Roderic very, very much for the beautiful lecture. And I remind you that the next lecture is this afternoon at 3, Professor Cedric Nocredam. So I will open the room a quarter of an hour before, and I invite you to attend. Thank you very much. And
Thanks,
thank you, Roderic.
Thank you very much, Roderic, for the patience and for being with us.
Yeah, and I hope that next year I can see you in the Café de la Corte.
Yes, I hope so.
Okay.
Okay, thank you.
Bye-bye.
Have a nice time.
Bye.
Bye-bye. Ciao.
Thank you.